1962, James Watson and Francis Crick won the Nobel Prize for Medicine for deciphering the structure of DNA, the molecule that contains an organism's genetic blueprint. Through experiments, Watson and Crick had revealed that DNA was a double helix polynucleotide molecule, with each nucleotide consisting of the deoxyribose sugar, phosphoric acid, and nitrogenous bases adenine, guanine, cytosine, and thymine that are commonly called nucleotides. After the DNA structure was revealed, these scientists got busy finding an answer to the next question: how the nucleotides get converted into amino acids that synthesized proteins, the building blocks of life. These scientists already knew that DNA directs RNA synthesis, which in turn acts as a template in the synthesis of protein. They therefore suspected the presence of a genetic code, a set of rules in a gene that instructs a cell on how to synthesize a particular amino acid from the nucleotides of an RNA strand. The scientists now had only one aim: to decipher this genetic code. George Gamow, a Russian physicist, was the first to achieve some success in deciphering the code. He initially suggested that since the mRNA strand consists of only 4 types of nucleotides that code for the 20 amino acids in the human body, they need to combine to form a particular code. Through mathematical formulae, Gamow then proved that a permutation and combination of 3 nucleotides would be sufficient to make up a specific code for each of the 20 amino acids. The logic behind Gamow's postulation was easy to see. A single nucleotide, for instance, would code only 4 amino acids. Likewise, the combination and permutation of 2 nucleotides would result in codes for only 16 amino acids. However, the combination and permutation between 3 nucleotides would code for 64 amino acids, a little more than required. But nonetheless, Gamow stuck to his postulation. Though scientists were now aware that a combination of 3 nucleotides was all that was required to make a particular code, they had another question: which combination of nucleotides coded which amino acid? The answer to these questions came from research scientists Marshall Nirenberg and Heinrich Mattei. They created a free cell system by grinding E. coli bacteria using a mortar and pestle to obtain E. coli extracts. The E. coli was then added to a test tube containing everything needed for protein synthesis, including ribosomes and amino acids, to name a few. However, the test tube lacked an mRNA, without which protein synthesis was not possible. The scientists then added artificially synthesized mRNA consisting of only one base, uracil, in the free cell system. The E. coli extract in the free cell system was now able to produce a protein chain made of one repeating amino acid, phenylalanine, with the help of the Severo-Ochoa enzyme called polynucleotide phosphorylase that was present in the extract. This suggested triplet UUU codes for the amino acid phenylalanine. Nirenberg and Mattei had finally cracked the genetic code for at least one amino acid. Subsequently, Hargobind Khorana formulated a chemical method to produce well-defined nucleic acids, long strands of RNA with every nucleotide in the exact position. This helped determine the rest of the genetic code, which was expressed in the form of a chart. Today we know that the genetic code is an array of 64 codons, a sequence of 3 nucleotides on an RNA strand that encodes a specific amino acid during protein synthesis. Of the 64 codons, 3 codons UAA, also called ochre, UAG, also called amber, and UGA, also called opal, act as a stop codon, which means they terminate the translation process and as such don't code any amino acid. Hence, they are known as nonsense codons or termination codons. The rest of the 61 codons code the 20 amino acids found in the human body. Moreover, one codon codes for only one specific amino acid, which makes the genetic code unambiguous and specific. For example, the codon UGG codes for tryptophan, and AUG codes for methionine as well as acts as a start codon, which means it initiates the protein synthesis. The genetic code is also a degenerate, which means that many different codons encode a particular amino acid. Take the case of serine, which is coded by 6 different codons including UCU, UCC, UCA, UCG, AGU, and AGC. Phenylalanine, on the other hand, is coded by codons UUU and UUC. Did you know that the genetic code is almost universal and the same throughout all living organisms? The codon GUG, for instance, codes for amino acid valine in plants as well as animals.